Bacteria can be found everywhere, from the fur of cute cuddly doggos and puppers to the screens of the mobile phones we constantly use. Some are harmless, like the thousands of airborne microbes we breathe in and out every day. Some are good, like the bacteria found inside our gut that help break down food. And some of them make us sick. Really, really sick. So what exactly makes these bacteria cause disease? What made these bacteria turn over to the dark side? This is the question a certain Filipino researcher in Australia is trying to answer. Welcome to this edition of Tea Minute Thesis.
Hey there, I'm Cami and this is today's edition of Tea Minute Thesis. I'm currently here with Brandon C., who is a PhD student at the University of New South Wales. So Brandon, how long have you been here in Australia?
I've been in Australia for about 2 years now, more or less.
So what did you study before coming here?
I did my bachelor's in molecular biology and biotechnology in the University of the Philippines.
Okay, and then what's your lab right now?
So our lab is a medical microbiology lab really right now. We study different kinds of pathogenic bacteria. Specifically, the way we like to look at them is that we like to look at the genetic basis for their disease and try to find ways to sort of counter that.
Within our gut is a thriving community of diverse bacteria that help further break down food into nutrients that can be absorbed by the body, and also help protect us from other harmful bacteria. One such mainstay in our gut is Escherichia coli, or E. coli for short. Inside the gut, E. coli is harmless, and surprisingly, outside as well. In fact, because of its relatively harmless nature and ease of handling, E. coli has a long history of widespread use in molecular biology experiments. If E. coli is so safe, then why do we occasionally encounter horror stories of E. coli causing outbreaks of food poisoning, resulting in massive product recalls? These outbreaks are actually caused by the more dangerous cousins of your average harmless gut E. coli. One cousin, known as enterohemorrhagic E. coli or EHEC, is particularly threatening. As indicated by its name, consuming EHEC in contaminated food could lead to bloody diarrhea and even kidney failure and eventual death. So why and how is EHEC so much more dangerous than gut E. coli? Their genome sizes or DNA lengths may provide a clue. Gut E. coli has a genome size of 4.5 megabase pairs, but EHEC had acquired an extra 1.5 megabase pairs of DNA through the movement of genetic elements among bacteria over the course of its evolution. This extra 1.5 megabase pairs of DNA gave EHEC the ability to cause disease, as genes within would encode for toxic proteins that could be injected into the cells of our intestine. But there's still much about the extra DNA that we don't know. Just like how computers require new software to control new hardware, new bacterial software is required to control the toxic extra DNA. And it's Brandon's job to uncover the workings of this new bacterial software, in hopes of providing some insight on how good bacteria can sometimes go bad.
Thank you so much for sharing your thesis with me.
No worries.
And that's it for today. So I'll see you next time for the new edition of Tea Minute Thesis.
Bye.